Okay, so we are going to look at Chapter 13, Characterizing and Classifying Viruses, Viroids, and Prions. And this is your list of study objectives. In the module, I will have questions that really go at understanding these study objectives. So my advice to you is while you're listening to the lecture, take very careful notes, pause if you need to, write things down. And then when you get to the end of the recorded lectures, then you can answer the questions without having to re-listen to the lectures again. So to save time, listen very carefully, write down careful notes, and then answer the questions at the end of the module. And that's what I'm asking you to do for this chapter. So viruses, we've talked about bacteria quite a bit, and we've talked about some of the eukaryotic cells that can cause disease, in particular some protists, protozoans. We've talked about some worms in lab. So what we're going to look at in this chapter is an overview of viruses. You can take an entire course just on viruses. The study of viruses is called virology, and you can be a virologist, so another expertise area within microbiology. And viruses are really interesting. They are not cells, and they are not considered alive, which is weird because they certainly have an effect on living organisms and living cells. So a virus fundamentally is a chromosome, and that chromosome can be made up of DNA or RNA, but there is no cytosol, there is no plasma membrane, there are no organelles. This is not a cell. Basically, a virus is a chromosome surrounded by a protein protective layer or compartment. And then some viruses also, in addition to that, have a membrane, but we don't call it a plasma membrane. We call it the viral envelope. And that word envelope, you've seen that before when we talk about the nuclear envelope. That's the membrane that surrounds the nucleus. So the viral envelope. But it's a plasma, it's a phospholipid bilayer, and it has proteins in it. But it's not, technically would not be called a plasma membrane. So the most simple viruses are, like I said, just a chromosome surrounded by a protein compartment, and we call that protein compartment the capsid, C-A-P-S-I-D. And like I said, so that's fundamentally what every virus has. And then in addition, some viruses have an additional level of protection we call the envelope. And what viruses do is they attach to cells and they enter cells. They go inside of the cells. And we saw that occasionally with some of the other microorganisms that we saw. Most bacteria don't enter your cells, but there's a few that do. But remember, there's a protist, Plasmodium, that goes into your cells. So it's not that unusual, but viruses, as a rule, the rule for viruses is that they must go into your cells to cause their infection. Now, their infection is just their reproduction. They are just trying to reproduce, and that was the rule of every organism. It's just all the things that it does is just an effort to reproduce. So it's not that difficult. Well, comparatively, it's easier to treat a bacterial disease than it is a virus. Viruses are really hard to deal with for a lot of reasons that we will cover in this chapter, but they are unusual. They are not cells. They are not considered alive, but they enter your cells or whatever host they target. And viruses are very specific. They target certain organisms, and every organism that I'm aware of has viruses that attack it. So it's very, I guess, fair in the sense that even bacteria can be infected with their own types of viruses. But a virus that attacks a bacterial cell would have no effect on your cell. So the viruses are very specific to their host cells. So viruses, like I said, are not considered alive. They don't carry out any metabolism because they don't have cytosol, they don't have organelles, they don't have structures inside of them other than just the chromosome. And they don't get larger, they just reproduce. But they can't reproduce without using a cell's organelles and enzymes and nutrients. So viruses most of the time have what we call an extracellular state, meaning an extracellular version of the virus and an intracellular version of the virus. So when the virus is outside of a cell, we call that extracellular, and when a virus has entered the cell, we call that intracellular. So the virus, when it hasn't gone into your cell, we call that the extracellular state. Once it's inside your cell, then it's referred to as being in the intracellular state. So in the extracellular state, we call the virus a particle, a viral particle, or a virion. So you pronounce that virion, V-I-R-I-O-N. Now all viruses have a protein coat surrounding their chromosome, which is made up of DNA or RNA. The protein coat is called the capsid. So this is some of your vocabulary that I was mentioning in some of my emails that I've been sending to you. The vocabulary is very important. If you can master the vocabulary, then you really have almost automatically an understanding of a lot of the material from the chapter. And I know you guys are good at doing vocabulary, so focus on that quite a bit. So like I said, some virus particles also have a phospholipid envelope, which is just a phospholipid bilayer. We just don't call it the plasma membrane. In this case, we call it the envelope. Within the envelope, I don't know why it did that. Within the envelope, you have some proteins that are used to recognize the host and bind, attach to the host cells. And once the virus attaches to the host cells, then there's several ways it can enter the host cell depending on the virus. Each virus follows a certain rule. And once it enters the cell through one of these ways, then we call it the intracellular state of the virus. Really what enters the virus or what ends up in the cell to cause the infection is just the chromosome. So as part of the entry of the virus into the cell, the envelope, if it's there, is removed, the capsid is removed, and you have just the chromosome is really what's entering the cell. And this picture shows on the top, you can see an artist schematic drawing of what an example of a virus could be. Viruses have different shapes. This one is called icosahedral in this picture, and the capsid is shown as these little pink circles, and each one of these is a protein that assembles into this really very geometric structure, but in the inside you can see the chromosome. The chromosome, depending on what kind of virus we're talking about, could be DNA or RNA. And there are single-stranded DNA viruses and there are double-stranded RNA viruses, so there's really a lot of variation in how the chromosome is structured. Down here, there's a TEM, transmission electron micrograph, that's been colorized, showing the surface of one of these sides of a virus particle. So like I said, the chromosome, the genetic material in the virus, can be DNA, RNA, double-stranded, or single-stranded. We've never found a virus that had both types of nucleic acid inside the same capsid. The chromosome is sometimes linear and sometimes circular. So that depends on which exact virus you're talking about, which one it would be. And a lot of times the chromosome is just a single molecule, but there are some, for example, flu virus that has many segments or many chromosomes. So we call that segmented when there's many separate strands of nucleic acid. A lot of viruses, though, have just a single chromosome. Now, virus chromosomes are very small. They're much smaller than the chromosome even in a bacterial cell. Viruses are very specific to their hosts. And some are even so specific that they will only attach to certain cells within the host. There are some viruses that are a little more general. We call them generalists. I would say that's less common. Most viruses are very specific. They're specialists. And like I said earlier, I think it's kind of fair that all organisms can be infected by their own types of viruses. Now this picture shows you a scale of size. It shows on the top left corner here you have a red blood cell. It has the typical concave center. You see the size, 10,000 nanometers, all right, which is about 10 micrometers. A bacterial cell is a couple micrometers, so you can see here a red blood cell next to a bacterial cell. Now guys, red blood cell is one of the smallest of the human cells. So 10 micrometers is actually really small for a human cell, but blood cells are pretty small. And then bacteria, by comparison, are even smaller. And then you can see in this picture on the top left corner here, you can see this little pile of dust right here, but they zoom in, so move to the right, they zoom in on that bacterial cell, and they're showing you E. coli, which has the flagella all over. You guys remember peritrichus. And then next to it are some viruses. Now, smallpox is a human virus. It's actually one of the largest viruses, so the fact that you can see it at all in this picture is kind of amazing. This little guy that looks like a little spaceship is the T4 phage, which is a virus that attacks bacteria specifically. So it's actually pretty reasonably large. And then down here you have some teeny tiny viruses, which we need to go to the bottom picture to see them a little more clearly. You have the polio virus. You probably heard of polio. There's another phage. Again, a phage is a virus that only attacks bacterial cells. So polio virus infects humans, for example, but phage only infects bacteria. So, but the sizes are pretty similar. And then over here they show you a tobacco mosaic virus, which has this very long elongated structure, and that infects tobacco plants. And you can see, just for size comparison, they're kind of highlighting on the inside of the bacterial cell how big the ribosomes of a bacterial cell would be. So on the order of the same size as an entire poliovirus particle. So these are tiny, tiny, tiny. So I like to, I'm very visual, I'm very spatial, and so I like to be able to figure out the size relationships for some of these things that we're learning about. So a relatively small human cell, which is the blood cell, is much larger than an E. coli, which is much larger than any of these viruses. So the capsid, if the virus only has a capsid, does not have an envelope, then the capsid would be how the virus would attach to the host cell. If the virus has an extra layer on top of the capsid, which would be the envelope, then the envelope would be the thing that would attach to the host cells. So whatever is the outermost layer of the virus, that's what attaches to the host cell. And something on that virus has to specifically recognize something on the host cells. So it's a protein-to-protein attachment. It's almost like if you think about when you learned about enzymes and their substrates, the substrate fits into the active site of the enzyme like a lock and a key. Well, these proteins on the virus fit into the proteins on the surface of the host cells in a similar way, like a lock and a key. And so when that key goes into that lock, it attaches that virus to the cell. And there's nothing the host cell can do. The host cell doesn't know that it should not be attaching to this virus. So viruses have co-evolved with their hosts to have this ability to attach to their host cells. They're very well evolved for that. Now, in terms of shapes of the virus, there's three basic shapes. Helical, which would be kind of like that tobacco mosaic virus we saw on the last slide, almost like a drinking straw, so very long and thin. And polyhedral would be one of these almost shaped like a diamond that's been cut with many facets. And then complex is just the way we say it when we don't have a geometric term to describe the shape. We call it complex. Sometimes complex is a little bit ambiguous, and we use that term when we don't know how to describe something. You'll see. So this picture here on the part A, all right, that is helical. So like a drinking straw. That's the tobacco mosaic virus. Most viruses are more of this polyhedral, which is shown in B, although you can't see it super clear in B. But let me go back to this picture here. On the polio virus or the bacteria phage, MS2, you can see that polyhedral. Bacteria phage T4 is interesting because it has a polyhedral part and it has a helical part. So it's kind of we would call that one complex probably because it has multiple shapes. So, but these are just words that we use as reasonable humans to describe the shape. When you look at something, you just describe it, what you think the shape is most closely like. These viruses on part D are more complex. I would call them maybe a bullet shape. So we just use words that make sense to us, but. All right, so the T4 phage, if you look at it, it is the most interesting and the weirdest looking thing. Whenever I look at this T4 phage, I think of in the first Toy Story movie, there was a doll's head on like a spider body that was like running around. I don't know if you remember Toy Story, the first one, but that's what this thing reminds me of. Now, of course, it's much smaller. But the chromosome would be up here, and we call this part up here the head. And then this helical part here we call the tail and the tail fibers. And this guy literally lands on the surface of the bacterial cell that it's going to infect. And the head crumples and pushes the DNA from the head. When the head crumples, it's almost like squashing a turkey baster. The DNA squishes and goes down through the tail. There's a hollow part in that tail part here, and it goes and it injects into the host cell. So the host cell is the one that's getting infected. In this case, a bacterial cell. So E. coli can be infected by bacteria phage. For viruses that have an envelope, the envelope will be on top of or surrounding, the capsid. So in this picture, there's a chromosome inside of a helical capsid, and then there's an envelope around that. So when you see that under a microscope, you can't tell the shape of the capsid because the envelope is typically it's always going to end up being a round blob surrounding the capsid, so you can't see the shape of the capsid in that case. So the enveloped viruses are always round, spherical. And it's almost like a little vesicle is really what it is, and the little vesicle contains the capsid proteins, and the capsid proteins contain the chromosome inside. So we say that the viral envelope is provided by, but I think maybe it's taken from the host cell during the very last stage of the viral replication cycle, which is sometimes called release. And that means release of the new virus particles. The viral envelope is made up of a phospholipid bilayer and proteins. In a lot of ways, it is the plasma membrane, but when it's part of just a virus particle, we don't call it a plasma membrane. But since it was taken from the host cell, it really has the same structure as a plasma membrane. What's funny about this is that the envelope viruses are more fragile, easier to kill. Well, they're not really alive, but easier to destroy the structure than a naked virus. A naked virus would be one that has no envelope. So write this down. All viruses have a capsid, but for a naked virus, that's all they have. Just the capsid with the chromosome on the inside. An envelope virus has the envelope, and then inside the envelope is the capsid, and inside the capsid is the chromosome. And it's funny, though, that the envelope viruses are actually easier to destroy than the ones that don't have the envelope. That seems counterintuitive, but it's actually pretty easy to disrupt a membrane because it's amphipathic, that soap detergents will typically disrupt the membrane. So, and there's other things, but they're actually a little bit easier to deal with. And we had in one, I think we had in one lecture how the envelope viruses are the among the least dangerous of the microbes, typically. Well, that's very typically, because I will tell you that the coronavirus is enveloped. But typically those are easier to deal with. Now, human viruses are really split into two families. We use the term families. And the two families that I want you to pay attention to, I mean, you don't need to memorize all these names. These are the names of the families. I will tell you that this suffix, or this part here, viridae, V-I-R-I-D-A-E, which is on all of these, that means virus family. So you have the pox viridae, that means the pox virus family. You have the herpes viridae, that's the herpes virus family, and et cetera, et cetera. The papilloma virus family, the polyoma virus family. So that viridae means virus family. But the big groupings, the big division of viruses fundamentally goes whether or not they have DNA chromosomes or do they have RNA chromosomes. So that's the big division. That's the two big family divisions. So with bacteria, we would argue that your big division of bacteria would be gram-positive and gram-negative. And y'all are familiar with that at this point. So what I'm saying is, in viruses, the biggest division point is whether it's a DNA chromosome or an RNA chromosome. I mean, yes, we're interested in whether it's enveloped or non-enveloped, but the DNA and the RNA is the main division that they have used as the first division of organization of viruses into classification groups. All right. So you're not going to memorize all these. Okay. All right, but you do need to know that DNA versus RNA is the main division point for classifying viruses. Now, when a virus replicates, it's going to enter a host cell, and it's going to use the host cell's own organelles and enzymes to make a lot of new virions. So reproduction is exponential, it's logarithmic, but it's much more logarithmic, it's much faster than bacterial reproduction. So you remember that bacteria can reproduce every 20 or 30 minutes, whatever the generation time is, but when they reproduce, they only make two. You start with one bacterial cell, and at the end of 30 minutes, you might have two, and then at the end of another 30 minutes, you'll have 4, and then 8, and then 16. It doubles each generation time. Virus is not just doubles, but usually it's like 1,000 new viruses are produced with each generation time. So one virus enters a host cell, and after 30 minutes, you could have 1,000 viruses that have been made from that one. So it is a much steeper logarithmic increase for every generation time that passes. So it's it's much faster reproduction than bacterial cells. I'm going to take a break here, and so that you can view the lecture in smallish modules, I'm going to stop and start up a new lecture module starting with this slide for part 2.